Hi and welcome to my Biocolour story called Co-Crafting Colors, which is a part of my practice-led PhD project combining biology and design. Today I will show you some of my work together with soil-dwelling bacteria. My name is Louise Hartvigsen and I am a doctoral student at Design School Kolding in Denmark at Lab for Sustainability and Design. I have been interested in biology for many years, and especially microorganisms and their capability of synthesizing different outcomes. This part of the PhD project is situated in Kolding, located in the southern part of Denmark as shown on the map. A central term to this project is localism, and since the project takes its starting point from the local soil in the surroundings of Kolding. I have searched for bacteria to try and find out where they live. I have taken samples from soil, grass, wood, rocks and pavements, all surfaces in my local environment, as you can see on the pictures. Here I have taken samples from the soil and spread it onto a petri dish prepared with nutrients for the bacteria to grow on the plate. Afterwards, I took the samples with me to the biolab I have at school and grew them at room temperature. This is how the different collected bacteria samples look, not bursting with color, but showing a variety of different microorganisms starting to grow on the plates. At a natural science lab, I got help to screen the genetic code of interesting bacterial colonies to be sure they were safe for me to work with. I present to you here the 5 different soil bacteria I am collaborating with at the moment. They are all producing colored pigments via different chemical signal pathways. In the following few slides, I will present each bacteria species for you. The first bacteria is Pseudoclavibacter, and it is producing a carotenoid pigment, which is very sharp and almost neon yellow. Here you see Rhodococcus, also a carotenoid producing bacteria. It produces a yellow pigment as well, but it has a warmth to it compared to the one I just showed you. Here you see Shingomonas, also a carotenoid producing bacteria. It is orange, as we know it from carrots, and has a deep warmth to it, and very easy to grow in the lab. Here you see Serratia marcescens, a proteodyosine producing bacteria. It produces a pink pigment, looking quite different from the red color as seen on the colonies as shown here in the picture. The last bacteria to present for you is Xanthinobacteria lividum, which produces a purple colored pigment called violacine. It has a distinct soily smell, and it can be quite tricky to grow at the DIY lab. On the picture, you see Design School Kolding. A design school which is built on the principles of Bauhaus, having open workshops where the students can come and work on their projects. This is also where I have set up a small DIY lab to carry out some small-scale experiments. Here you see the DIY biolab which I have set up at the basement at the design school. Here I can grow my bacteria in my polystyrene incubators and store them in a fridge that I have especially for the bacteria. Working together with the bacteria in the DIY lab requires patience and time to get to know the bacteria. As you can see on the left side, it went well growing the bacteria and getting them to produce pigment. In the picture in the middle, the bacteria did not produce pigment, although they had the same growth conditions. The pigments have been applied using different application approaches, growing the bacteria directly on the textile, stamping the pigments onto the textile, and using the pigments to dye the textiles with a full surface color. Some of the samples have been exposed to sunlight to see how fast the color fades and also look at how the color fades, revealing the nature behind the pigments. As Tolman states, rather than designing static expression, different lifespans could be embedded in the textiles through choice of material and construction, enabling the designer to tailor lifespans to both the object and textile it is made of. Considering how a textile's expression can evolve during the design process opens up the possibility of achieving many expressions from one starting point, challenging how aging and use are valued. This is also something that I have tested with this experiment to see how the color fades when exposed to light. Maybe we as humans are ready to adapt our behavior and accept changes in a textile material as something beautiful and valuable, hence appreciating nature and its natural processes, also challenging the designer's process to design with a holistic mindset. Thank you.